We're really excited that you're here to talk to us today, and your talk is the mechanical basis of memory. Do memories reside in synaptic scaffolds? So I, for one, am very interested in hearing about this, and I will turn the floor over to you, Ben. Thank you again for coming.
Thank you, Melissa. Thank you for the nice introduction as well. Very comprehensive. That was good. Time flies. So yeah, as Melissa mentioned, I've had a circuitous career, but since 2006, when I moved to Leicester to work with David Critchley, I've worked essentially on this same thing, trying to understand what this guy in the middle, shown here, this is a protein called tailin, and over the years what we think it's doing has evolved, and as you find out more, you realize more about what's going on. But what I'd like to try and convince you today is some of the discussion of how this is a really elaborate mechanical sensor, and how it can also store information inside of its shape, and it can change shape a lot to enable it to give lots of different signals, and ultimately to how expanding this from having a molecule which can store memory, it's got molecular memory, to how that could potentially fit with a concept of a mechanical basis of memory. So this talk is going to be squarely in two halves, really. It's going to be the first half, which is going to be experimental, how we've got to where we are with our understanding of this molecule, integrin signaling and mechanical signaling. And then the second half, to rethink about this and rethink about what might be going on in the context of an organism, to end with some speculative a theory I'm in the process of developing, this so-called MeshCODE theory. And hopefully I could convince you, or at least open a discussion, which I'd love to get feedback on, as to whether what we're talking about here would suggest that memories might reside in the synaptic scaffold. So this image here is just I find it hypnotic to watch. It's good when I'm trying to get, it's like a binary coding. I'd like to argue that this is a binary coding which we've been looking at all of these years, but it's only in the last year or two it's really understood this. So my focus when I started in 2006, and the main focus what we do is on these interactions which animal cells make to the extracellular matrix. This is a complex meshwork of proteins and other factors which all of our cells attach to. And this slide I've always shown since 2006. It's old, you can see from the imaging, but it just to highlight how essential this adhesion is to multicellularity. It's fundamental to the development of multicellular life because it's involved in all of these different processes and many more. And most of these cell matrix interactions which we're interested in, and our collaboration with Melissa are interested in, is the ones mediated with the Integrin family of ECM receptors. And this cell shown here is spread out, and these green assemblies around the edges where this cell is physically making adhesive structures to an underlying extracellular matrix. And I'll just draw your attention to these really strong stress fibers which connect all of these together. These are mechanical linkages. They require coupling of the receptor and the ECM to the cytoskeleton to be able to form, but that also enables them to be able to feel as well. And that's what I'd like to talk to you about now. I'd say that more recently, one of the things which has really come to the fore, and one of the things we're very interested in, is this ability of a cell to respond to its physical environment to lead to changes in its gene expression and its fate and how it differentiates. And that's probably best shown by this experiment from Dennis Disher's lab about 15 years ago now, probably a bit longer. And it's a really simple experiment, but it really lays out what we're talking about here, where you can take a population of undifferentiated stem cells and you can differentiate them into all of these different lineages, depending on what signals you give. But the smart thing about this experiment is that the chemical and the growth factor and the serum signals are all identical, and also the ECM substrate that this is on is identical. The only thing that they're varying in this substrate is the physicality of it, the elasticity. But just by changing how stiff this substrate is, these little blue squares, which are the adhesions, sense that, it feels it, and then that leads to this reprogramming, to the point that if you put it on a really soft substrate, you can make a fat cell or a brain cell. if you put it on glass, you can get it to more like a muscle or a bone cell, purely by changing the physical environment. Obviously, in our bodies, there's a lot of other signals and a lot of other factors which come together. But in this rawest of experiments, the only variable here is the stiffness. So what this tells us, and what we're very interested in, is that these adhesions, these tiny blue squares, are clearly information processing centers, because they're feeling that environment and then using that to change the whole behavior of the cell. And if we just go down into a little bit more detail on these blue squares, then these blue squares are actually large multi-protein complexes, shown in this very nice sweetie diagram from Tony Kanchanawong and Claire Waterman, where here we've got the integrins at the bottom of this cell, binding to this blue, which is the extracellular matrix. So hundreds of these integrins make these attachments, and then they couple to the actin stress fibers and the actin cytoskeleton, shown in blue. And what we work on is this connection between the two, between the integrins connecting to the outside world and the cytoskeleton, which is the cell's contractile machinery. And it's so-called this integrin signaling force transduction layers. And to just go into a little bit more detail on what that looks like, what we've got is on the surface of our cells, we've got these receptors for the ECM called the integrins, and they're shown cartoon form here, and they're shown grabbing onto this extracellular matrix at the top of the page. And one of the striking things about this adhesion is that the integrins can adopt different conformations, and they can be shut down by folding up like a fist to prevent engagement with the extracellular matrix. And they can become activated to unfold and grab on. So the cell can coordinate whether it attaches or lets go. And this is very important during migration, but it can be controlled from within the cell. And one of the main regulators, in conjunction with kindlins, is the protein tailin. And that's what we work on. That's what the focus of today's talk is going to be on, is this large rod-like molecule, which is in the cytoplasmic, and it can pull on these tiny little tails of the integrin like a puppet master, and in doing so, coordinate the activity state of the integrin. So in the simplest sense, tailin and kindlin pulling on these tails will switch the integrin on, and reciprocally, them letting go will cause it to let go. But what tailin also does, and if you remember in that first slide, it makes this important linkage because it couples the matrix and the integrins to the cytoskeleton, and it does this by a number of attachment points. So tailin's an essential linker in all of these integrin adhesion nearly all of these integrin adhesion structures. And what's become apparent is that tailin, being positioned here, it's perfectly positioned to be able to sense and respond to mechanical forces, both from outside the cell if there's a perturbation or there's movement or it's changing environment, and also from inside the cell where the cytoskeleton can generate forces. And this guy sat in the middle is perfectly positioned to respond to these changes in force. And this has led to this notion that tailin's a mechanosensor. And the canonical tailin mechanosensitive event is that when you stretch this system, you expose binding sites for a protein called vinculin. And then vinculin is recruited and it cross-links tailin to actin, reinforcing these adhesions to make them big and strong. And in essence, that's the core of nearly all of these integrin adhesion complexes. You've got an integrin, a tailin, kindlin's not drawn, a vinculin, and actin. But onto that core of five proteins, you can assemble hundreds and hundreds of different proteins to give rise to these many different diverse structures which are mediated via these integrin adhesion complexes. And two of them are shown by these beautiful videos from our collaborators, Mark Morgan and Guy Tannenzapf. And here we've got in green, we can see these adhesion complexes, and this is a fibroblast crawling along a surface. And you see these large complexes form at the front, assemble, and then contract and pull towards them, and then disassemble at the back. Highly regulated signaling complexes, which also enable the cell to crawl. That same machinery, those same four core components, are essential in these linkages holding our myotendinous junctions together. This is a developing Drosophila embryo. And Also, you've got all of these weird and wonderful adhesive structures like podosomes, invadopodia, and many, many others. All of them assemble on that core component of an integrin, a tailin, and an actin. And what I can hopefully convince you over the next bit of time is that tailin can adopt the reason that these three proteins can adopt all of these different functions is that tailin can adopt numerous different shapes, different conformational states, which run different programs. And lately, one of the things which we've become very interested in the lab is the adhesions, integrin-dependent adhesions, which are involved in holding these synapses together, both in all of the different synaptic junctions. These all absolutely require ECM, they require integrins, and they require actin, cytoskeleton, and regulation. So we're very interested in these, and I'll talk about these more in the second part of my talk. But just to go a little historical trip through how we got to this understanding of this molecule, when I was first hired at Leicester in 2006, tailin was known to be important in these linkages, but it was perceived to be mainly just an adapter protein, which could reinforce these and couple integrins to the actin cytoskeleton. And it was known there were 62 helices, but very little was known beyond that. So trying to get at what this molecule was doing with this level of detail was not trivial. So this next animation summarizes about 4 or 5 years of work. But to get to those 62 helices, what we were able to do as part of a collaborative effort in Dave's lab was to work out that there's 13 of these rod domains. So these 62 helices arrange into these 13 helical bundles. And to a first approximation, these look very boring and monotonous. I actually spoke about this at a repetitive molecules conference one time, but it's hopefully I'll show you that these are all unique in their own right. So what we've got in essence, we've got an integrin tailin matrix connection at one end. So this molecule sticks onto a relatively firm surface, and then it couples to the actin cytoskeleton at the other end, positioning all of these 13 domains in this force transmission pathway, meaning that all of these can experience forces to different levels when this molecule wires up and when it starts to feel contraction during the cellular processes. And at the time doing this, we were considering these to just be folded globular domains. This is the main thing we saw. But one of the big breakthrough moments which changed my whole direction of travel was in 2012, when we discovered that one of these proteins was binding to 2 different ligands. And I'm going to show that on the next slide. But just to summarize, what we could see was that this domain, it was R3, the first switch we identified, exists in this folded conformation shown here, but it can change its shape to open to this extended conformation. And these two, it looks like, oh, it's just unfolded, but the striking thing here is that this is reversible, and that if this is maintained in a force dependent linkage, if it's held at one end, at both ends, then both of these conformations are thermodynamically stable. So it will stay in the conformation it's in until you change it to a different conformation. So this means that what we've got is a switch. We didn't realize this when we first started, but it's now become apparent that this is a switch. It can be in this state, and it can be transitioned to this state, and it can transition back. And we didn't just think, oh, these domains might unfold. The reason for that came, as I just mentioned, was our biochemical characterization of how tailin interacts with different ligands. And we were very interested in a RAP1, a small GTPase, its effector molecule, RIAM, and it was shown to be important for tailin activation and integrin activation by Mark Ginsberg's lab. So what we were doing some biochemistry, and what we found was that RIAM binds to this folded switch domain. So I've drawn the RIAM helix, which interacts the epitope as a green cylinder. It sits on the side of this domain. And this was took us a while to appreciate what this shows, but this was a real change of direction, made me into a mechanobiologist overnight from a biochemist. Because here we've got this domain, R3, binding to a helix of the protein RIAM. But what was already known was that this domain unfolds to expose this red vinculin binding site. So here we've got the same domain of tailin binding to 2 completely different ligands, coupling to 2 different signaling pathways. And it can be driven by mechanical force. That force exerted onto tailin can transition between this domain, this conformation, and this conformation. So your drivers switch to the vinculin tailin R3 complex and disrupt this. And we've done a lot of measurements on this and a lot of theoretical studies. And the transition in terms of the binding constant, the KD, is greater than 10,000 times across this gap, to the point that RIAM won't bind to this complex at all, and vinculin struggles to bind to this one. So what that means is we can now see a way that the cell can convert forces, mechanical signals, into biological signals. You change the shape of a molecule, and in doing so, you change the signaling up through that molecule and into the whole cell. So this was super exciting to us, and as soon as I got this, I got in touch with Mike Sheets at Singapore. There's a mechanobiology institute, and I said about this switch, and I wanted to know how much force it would require to convert from this state to this state, how hard you'd have to pull on tailin to flick this switch. And he put me in touch with a longtime friend and collaborator, Ji Yan, and his excellent postdoc, Ming-Chi Yao. And this collaboration has been incredibly good fun, and we've learned so much beyond what I'd ever think we would ever know about this molecule. And the first experiment we wanted to do was to know how hard you'd have to pull on tailin to cause this R3 domain to unfold. And the experiment which we did here, which is a few years ago now, is conceptually very simple, but mind-blowing that it's possible to do this. But what we can do is we can take a tailin molecule and we can covalently attach it to the cover slip of a microscope via engineering the N-terminus. And then via the C-terminus, we can again engineer it so we can tether it to a magnetic bead. And what that means is that we've now got a magnetic bead tethered to a surface via our protein of interest. And then we can use a magnet to pull on this bead, a tiny little permanent magnet, and pull on this bead, and we can measure how hard we're pulled and how far we're pulled. And we can then look at how the protein responds and how its interactions with other ligands respond. And I'm not going to show I'm only going to show 2 slides. No, I'm not going to show much mechanobiology data, but here's a typical data set which shows the complexity of the tailin mechanical response. So in this experiment, the first one we did, it was just pulling on 3 domains of tailin, R1, R2, and R3. And the experiment here is that we pull on the bead and we have increasing force. And then we measure the extension, we measure how far that bead is from the surface. And what you see is when you pull on a tailin molecule, it starts to get pulled taut, which isn't surprising because you're stretching it. But you see these transitions here, and there's 3 domains and there's 3 discrete transitions. So each of these transitions is one of these domains unfolding. And they all behave completely independently of each other. And this is also 6 cycles in the same experiment. So you can pull it until it's completely spaghetti-like, and the second you let go, it will immediately fold back up, and then you can start again. And in the absence of other factors, you could do this hundreds of times indefinitely. So this told us so much about what was going on. Each domain is behaving independently. Each has got its own mechanical response, unfolding at different amounts of force. And I'm not going to talk about it today, but we can start to make mutations to prove that this R3 domain is the initial unfolding response, because we can stabilize this domain and show that that first unfolding step shifts to the right. It needs a higher amount of force to flick that first switch. And again, I don't want to show too much detail here, but just to say that if you do this, you can repeat this experiment as many times as you want. It'll always give the same unfolding response. It's incredibly robust, and it's incredibly, well, it's just incredible that you can reset this thing so effectively. What's interesting is if you repeat this experiment in the presence of vinculin, and you'll recall that vinculin binds to that unfolded state. So if you repeat this experiment with no vinculin, you see this. If you repeat it with vinculin, then the first go, you still see the unfolding steps. But then after you've stretched it one time in the presence of vinculin, vinculin, because it binds to these domains, it stabilizes that folded state. So what's vinculin's doing here, as well as reinforcing the adhesions, is it's stabilizing the patterns of folded and unfolded domains, helping to lock them in that conformation. And just as an aside, what we can clearly see at high force in the presence of vinculin is we can see these small, tiny extensions in this profile. And you don't see those in the absence of vinculin. And these were head scratching for a minute, but what they correspond to is a vinculin binding site helix, as a helix shown here bound to vinculin, transitioning to a random coil kicking off the vinculin. So each one of these steps, which we can see in these profiles, is a single vinculin molecule getting kicked off of a single tailin molecule. So we can actually count how many molecules are talking to each other and interacting in this experiment. So that's enabled us to quantitate this and to also measure the force-dependent binding constants, etc. But what we're starting to do now, this is enabling us to get a detailed understanding of how these mechanical linkages between tailin and vinculin work, and how these patterns work, and etc., and how they respond to force. So this R3 domain, to summarize the very first bit of the talk, is that it turns out that this is a key checkpoint in adhesion assembly. So this is a good example of how these tailin switches function. Here we've got a cell which is crawling down towards the bottom right. And right at the very front of the cell, you start to get enrichment of this RAT1 effector called REAM. And REAM serves to bring tailin to the membrane. It brings so what happens is all of these components come together at the front, and then they unpack, and then they give rise to these adhesions. And you bring a tailin to the cell edge, and it meets an integrin. And when it meets an integrin, again, Kinley's not drawn on here, sorry, Melissa. But once it meets an integrin, it can start to activate that integrin. But at this stage, you can imagine that this molecule is not under any significant forces. It might be tethered at one end, but nothing's pulling on it at the other end. It's only when you successfully wire this thing up, so it's bound to an integrin ECM complex at one end and captures the actin cytoskeleton being generated in the retrograde flow at the other end, that this molecule is now tethered and it starts to experience mechanical forces. And that first pull on tailin is sufficient to unfold this R3 domain, displacing REAM and simultaneously bringing vinculin to hold this thing together and to make these strong connections. So this first switch in tailin is the on-off switch for making an adhesion. And I think this is really important because these complexes grow to be hundreds and hundreds of proteins big. You don't want these complexes just to start forming willy-nilly all around the cell. They only form at the right place and time once all of these different connections are made. And then this mechanical switch is the checkpoint. And then once that's happened, these things can grow and expand and more force can be exerted. And you end up with these really large vinculin-rich complexes. And this on-off switch coordinates that. So once we found that switch and we found that it could have such a striking effect on cell behavior, and we got better at cloning and better at the tweezer experiments, the obvious experiment to do was to look at the mechanical response of the whole of the protein, all 13 of these switches. And not least that was driven because in that meantime, we'd done a lot of proteomics on tailin, and we'd found that there's more than 40 different ligands which decorate these domains. So we wanted to know if all of these were switches. And not surprisingly, because I'm telling you it here, is that yes, indeed they are. This experiment here is still absolutely jaw-dropping to me even now. This is the same experiment I discussed previously, where we're increasing the force, but here we're stretching all 13 domains. So what we can clearly see is here's R3 unfolding. This is at low force. This is that initial switching on the adhesion. But once that's formed and the adhesion's formed, then each of these steps in this staircase is corresponding to one of these rod domains unfolding. And they all behave completely independently. So one unfolds, the whole thing doesn't fray like when you pull on a string on your jumper. It's one unfolds, and that's a quantized thing, and then you'd have to pull again, and then the next one would unfold. But because we can see that each of these domains is binding proteins in its folded and unfolded states, then each of these steps in this profile represents a change in the shape of the tailin and a change in the biological signaling running through that tailin and running through that adhesion. And where this gets interesting is if you look at what those 40 proteins look like and the different regulators which are decorating those mechanical switches, they essentially coordinate all walks of life in the cell. You've got the regulators of the cytoskeleton, GTPase effectors, transcription factors, channels, PIP kinases, cell cycle regulators, all of these different things all decorate depending on the pattern of those switches in that tailin molecule. And there's hundreds of those tailin molecules, so it's the overall ratio and it's the overall number of all of these different switches, which enables an incredibly graded and tuned signaling response through these adhesions. And what we now think is that each of these switches can coordinate a different cellular function. For instance, the R3 domain switches on adhesion maturation. And this would all, one of the things we got asked very early on was, this is all theoretically very good, but does it occur in a cell? If tailing is fully folded in a cell, then obviously that's not likely to be happening. But what we can clearly see is by measuring the end-to-end distance in inside a cell, tailing end-to-end distance, that at any time between 1 and 9 of these domains must be extended to give this extension of up to 400 nanometers. So what we can see is that this is highly diverse, but that depending on which adhesion structure you're in and what conditions, you have different amounts of these switches which are unfolded. And I'll skip through this quick because I want to get onto the interesting stuff, but just to point out that the classical view of tailing is a linker to between the extracellular matrix and the actin cytoskeleton. One of the things which we've become quite interested in is what these other switches do. And I just want to draw your attention to another one of these switches, the R7 domain here, because it's controlling microtubule targeting to adhesion sites. So the role of tailing coordinating actin cytoskeleton is really established. But what we can now see is that tailing's coordinating the microtubule cytoskeleton at these sites. And again, I don't want to go into this in too much detail, but just to say that when this R7 domain shown here is in an adhesion in a folded conformation, it can recruit a ligand called CANK, and you might come across CANK proteins if you're interested in adhesion microtubule dynamics. But when CANK binds to tailin, it creates another large complex, which means that microtubules, and you see those beautiful videos of comets and microtubules going outwards, they tend to target to the adhesion sites. And that's because this complex serves to capture those microtubules at these sites. So CANK is coordinating, tailin is coordinating the actin cytoskeleton and the microtubule cytoskeleton. So depending on the shape of the tailin molecules in those adhesions, you're recruiting different cytoskeletal components and making different mechanical linkages. And what's striking is we're biochemists, so we want to try and disrupt each of these complexes in turn. And what's really striking is that you can put a single point mutant into a tailin molecule in that R7 domain, and you can break this link to CANK, and you can prevent microtubules from targeting to adhesions at all. So this linkage is the direct way that microtubules target to focal adhesions. And I forgot this slide was in, but yeah, we can show that here. If you look at how microtubules behave in a cell, this is in collaboration with Anna Akmanova in Utrecht, then they grow out really fast, but as they get towards the cortex, they slow down, and they tend to be captured around the adhesion sites in the ones we're interested in. We pop that single point mutant into tailin, then you lose that control of microtubule dynamics. They grow really fast the whole time, and they don't slow down, and they're not captured, and they're incredibly dynamic and more unstable. They've got this instability. So tailin is, depending on the shape of the tailin at the front, is coordinating how the microtubules are laid out in the cell as well. So what this tells us is that mechanotransduction through this molecule is pretty complicated. And we can start to draw it more schematically with these multiple different attachment points, these different mechanical linkages, this connection to microtubules. And it's really, we've got all of those 13 switches, which can be in different shapes and different tailins, hundreds of different tailins, 40 or 50 different ligands. All of that is really complex, but somehow these complexes and all of these forces and all of these different inputs work. I mean, we form with incredible robustness. So what we can start to imagine is that we've got this complex signaling hub where we have a molecule, and depending on how it's pushed and pulled, the tailin adopts a different shape. And that shape of that molecule recruits a lot of different signals, which give that adhesion its unique signaling response. And it also helps to all of these to talk together and to work as a team. But as you increase the force, as things change, then you change the shape of these domains individually, but you're constantly changing and tuning the signaling on that molecule in that adhesion. And as you pull harder, you might unfold one domain, and then you might unfold another domain, and you're changing the color of these ligands, which would be different signaling molecules in that list of things decorating tailin I showed earlier. So we're now very interested in this tailin code as a conceptual framework for understanding mechanosignaling and mechanotransduction. And our hypothesis is that it regulates a wide range of cell functions through these conformational transitions that are modulating binding interactions, such that in different shapes, in different cells, in different conditions, different tissues, this forms a different pattern of these domains, which gives rise to a different signaling response, enabling the cell to instruct the cell how to behave. And because this is so complicated with this network of competitive interactions and 13 different switches and all these different cytoskeletal linkages, this means that it's really hard, but it's also got incredible power, and there's many different ways that you could arrange these adhesions. Even with just those 3 or 4 proteins in the core, they can adopt many different shapes and many different signaling complexes. So that's why we think there must be a code. This is robust. Every cell in our body knows what it's got to do and what it needs to do next. And a lot of that is via this mechanical grounding. It's this mechanical signaling responding to the environment. And that's what we're trying to understand, this mechanotransduction code. And that leads me into what I want to talk about now, because since we've been working about the tailing code, but in collaboration with Nick Browning in Cambridge, we've been looking at this more and more. And what you see is, if you look at what an adhesion looks like, it's not just individual tailing molecules. These proteins are arranging into like a meshwork where they're cross-linked with different other proteins like vinculin and other and the dimeric. So you end up with what is essentially a complex meshwork of these signaling mechanochemical switches. So what we think is on the bottom of the cell, you've essentially got this really sophisticated signaling machine, this mechano-signaling machine. And depending on the shape, depending on the environment, these form a different pattern, a different array of these switches. And then all of the ligands, which we all know and love, which are linked downstream of adhesion signaling, then decorate onto this in a context-dependent manner. So depending on the shape of the switches determines how many of these binding sites for each. So you end up with this really complex signaling downstream of integrin adhesion complexes. So what we think is that there's, on the bottom of the cell, there's this really extensive meshwork providing constant mechanical signaling. So each one of these would be a node in this meshwork, a tailing vinculin connection. And it's on the bottom of the cell, constantly feeling and sensing its environment. And you can ask a couple of simple questions to get how this might work. Is that you could ask what would happen if a force was applied onto this meshwork. And here's an image. It's actually gravitational field waves, but it's the same conceptual thing. Is that if you imagine this meshwork and you deform it with a perturbation, either damage or intentionally, or you change it, then all of those tailing vinculins in that site are going to experience that force much greater than these ones around the edge. And the bigger force is going to cause bigger changes. But what that means is you can change the pattern of these switches in those adhesions at that site and change the signaling from there. And this can enable the cell, if it's damaged and intentional, to repair or to change what it's doing, or the instructions that cell is receiving. So we think that tailing and vinculin and this meshwork is incredibly complex, and it's enabling this constant sensing and responding to its environment. And I just want to now just say, so for a long time you had a tailing and a vinculin, and that was a really simple, it was just a mechanical linkage. But now what hopefully I can show you is that with all of these vast array of switches, what you've got is the ability to have incredible diversity and nuance in this signaling response with just 2 different proteins at the core of this mechanical linkage. And this can be exemplified if you just ask 2 very straightforward questions. And both of them, I think, are true. But you could imagine, what if each tissue in our body has its own mechanical properties, and this gives rise to its own bespoke array of these switches in this mesh code? I think that is what happens. The physics of each different part of our body is very different. And also, if each tissue has its own proteome that has proteins that engage the mesh code differently. And again, each tissue has different genes expressed, different proteins produced. And here's just some slices of a brain and looking at what proteins are expressed. And you can see that there's huge differences in the relative levels. This is from, I think it was the Brain Atlas. But it's not hard to imagine that within these blue and red dashes, there might be things which either interact directly with the mesh code or interact downstream of that or upstream of it to give rise to this response. And if so, that means that this mesh code has the capacity for incredible diversity and nuance in this integrin signaling, way more than just grabbing onto an integrin and coupling, like a level which I don't think I can even comprehend at the minute. So this signaling layer here is incredibly powerful and can coordinate all cellular functions and input into all signaling pathways to some extent. So to be controversial, I did this initially to wind up Mark Ginsberg, but I think that this isn't an integrin signaling layer. This is a tailing or even a mesh code signaling layer. This is a mechanical computer which is instructing the cell how to behave. So to that's the actual experimental stuff. Now I just want to talk about a change in my thinking in how this could occur, which leads to what the title of the talk was about, how this might be a way to encode memories. So the classical view of tailing is a mechanosensor. You pull on it, it changes its shape. And in essence, in mechanotransduction, what you're envisaging, and sorry for the slightly naff guys in suits having a tug of war, but you've got this interplay between the extracellular forces, the movement and things changing outside the cell, and the cell's force generation machinery. And depending on these balance out, these pull and relative like tug of war, and these switches, these 13 tailing switches, sit in the middle of that, and they respond to changes from inside and outside the cell. And this enables constant force feedback and enables the behavior. What I came to, this was back in the day where you're allowed to travel and go to conferences. When I sat at an airport in Sydney, I started to think about this as a very simple question. But what happens if this extracellular environment is constant and predictable? And I'm showing that as a wall. What I'm not saying here is that this is stable and never changes, because I don't think that's the case. But in a lot of our cell types and in a lot of our tissues, the environment is incredibly predictable. It's constantly maintained. It's constantly repaired in such a way by the cell itself so that the cell knows what that is. So if this is a predictable thing, then here we've got a very different scenario. We're not, the switch pattern of the tailings isn't dependent on two varying things. What we've got here is that the extracellular forces can be largely known. I wouldn't say constant, but known. It's no longer a tug of war. Here what we've got is we've got a mechanical device attached at one end, which the cell can pull on and generate forces to cause these transitions and flick these switches internally. So here we've got a switch box which the cell itself can control to coordinate its function. And here, so yeah, the cell now generates forces and alters the switch patterns. So it was that finding, that theoretical shift in my thinking, which led to this paper, which has been under review since, I don't know, I was a young man when it first went in, it seems. But this is a theory I've been developing, trying to understand what would happen if this was predictable and if this could occur and be stable. And that's what I want to talk about for the last 10 minutes or so, is this theory and this idea to get feedback on this. And it's this, I mean, if the cell can coordinate this, then the cell, a signaling pathway, could trigger contractility, which would flick one switch. Because these switches are individual and they're quantized, then you could flick these individually and control their pattern via intercellular. So now how I start to think of tailing, and a lot of work we're doing at the single molecule and the theoretical level, is to consider that tailing and these mechanosensitive events I've been talking about, this is actually information which has been stored in these, because we talk about these domains going from a folded to an unfolded state. Both of these are thermodynamically stable, so you can switch between that to that with mechanical force, or reduce the force to go back to that. So if you think about this is actually a binary switch, then this is a folded state, which is O, but you can pull on that tailing and trigger it and convert one of these domains to a 1. And here's a small example. If you had 3 domains, you exert force on it, you would transition 1, and then the other 2 would stay folded. And then you could either reduce the force and go back to that, or you could pull further and unfold another one. But because each of these have got their own unique mechanical response, the order which these will unfold when you pull on it is quite reproducible. And because of the feedback, because of what's recruited, then this happens with incredible robustness. So what you could start to imagine is that what we're actually really looking at is a binary string, which in the the cytosol is all in this folded O state, which is what we did all of our structural work on. But once it couples to the integrins at the and makes these adhesion complexes and connects to the actin cytoskeleton, then they can adopt a certain pattern of these switches, which coordinates the cell how to behave. But from that position, you can then generate contractility. As say a signal generates an increase in RAC activity, or you generate contractility, you can convert one of these switches, changing this string of information, and you might pull again and change another. And then you might have a decrease in contraction, or you might have proteins diffusing away, which lead to a reduction, and then the pattern can change again. But all the while, this is information that is being stored in these molecules, and this is information which is coordinating the cell how to function. And I think this is what's happening in those differentiating stem cells. But I think it's information which is occurring. And the reason I think that, and what we can show, is we can show that tailin is an information storing molecule, because we can do single molecule studies on it. And because we now know the unfolding and refolding kinetics, we can simulate this in silico. But what this enables you to do is to hold a tailin molecule, and if you hold it under tension, then you can add numbers to it by pulling on it, or reduce numbers by letting a contraction. And as long as you hold both ends of this molecule, it will remember that information. It will remember the shape it's in indefinitely. And that means you can do sums on it, because it will remember what state it was already in, and then you can add more to it by pulling on it and doing these switches. A big assumption here is that this only works if the mechanical environment is perfect and you've got a perfect tether. If it's constantly moving, then that it's going to still store information, but it's going to be very noisy and not very persistent. But if it is predictable, and this leads to a prediction which arises from this theoretical thinking, is that the mechanical environment of the brain is perfect. And for a long time, mechanobiology in the brain and mechanical transduction, because the brain is soft, it's been considered not to be particularly effective for mechanotransduction. But this is actually a strength, because this means, because it's soft and because it's not that stiff, then that means that there's the opportunity, and the theory predicts that every single synapse in the brain will form its own extracellular matrix around the edge to form a perfect mechanical unit. And this is borne out because if you look at any sort of neuronal process, the ECM is constantly undergoing maintenance and remodeling all the time. And I think that's to make this a perfect mechanical environment. Because if the mechanical environment is perfect, the prediction is, and what I'm arguing here, is that mechanical computation can be perfect, and that you can generate these same responses every single time and with high fidelity. And the reason to go back to the strapline in the title is that if you look at what a synapse looks like, and apologies for my poor artwork, this looks like more like a nostril than a nose than a synapse. But if you look at how these things are organized, then you've got the active zone and all of the classical neuroscience, but you've got round the edge, you've got the ECM components, and you've got the integrins, and you've got the actin cytoskeleton. So the integrins, you don't normally get an integrin which hasn't got a tailin attached to it, because tailin regulates the integrin activity. But tailin's been neglected just as a linker. But if you look at the literature on integrin's role and ECM's role in pretty much all neuronal processes, it's quite well established. But what we've got, you can start to re-envisage this as a mechanical unit. Around the periphery, what you've got is you've got an array of switches in these scaffolds, shown cartoon here. And this, you've got to remember, this would be decorated with hundreds and hundreds of signaling molecules to give rise to the adhesion itself. And to show this slightly more conceptually, if you imagine that all of these switches, I've drawn them in this different array here with a tailin molecule with 13 switches running down the page, then in an adhesion, you might have 100 tailins. Then there'll be all of these different patterns of ones and O's, depending on whether each of these domains is folded or unfolded. And the hypothesis I'm generating here is that electrochemical transmission across this site causes a contraction. It's not, these things aren't under high force, they're under low force. That's how you store this information. But when you signal, you get contractility, a short burst of it, and that changes the pattern of these switches in that synapse on both the pre and the postsynaptic side, and potentially in any supporting roles as well. So as you signal, you could be constantly updating these switches to change and tune how each of these synapses behaves. And again, one of the things we're trying to do with Melissa is to establish how many of these molecules are actually in the synapse. I mean, it's not a given. Integrins are definitely essential and they're there, but how many? If you assume 100 tailings per synapse, and 8 of these switches are able to be used in this, then you might say there's 100 bytes of information could be written into these scaffolds. But what this means is the hypothesis I'm generating is that memory is mechanical in nature, that this binary coding of these mechanical switches which hold the cell hold the synapse together is actually where information is stored, and the electrical and chemical signaling and transmission across this updates this coding in both the pre and the postsynaptic sides, altering the information in that synapse, but also altering the activity of that synapse. And something which is incredibly speculative, even more than that, is that every single cell in our body, apart from the ones which aren't attached, but 99%, nearly all of the cells in our body absolutely require integrin adhesions. So every single cell has got this mechanical coding in it. So you could start to envisage that this is a mechanical code, a mechanical machine code, which coordinates this whole organism, any organism. But one of the things which is really important is that this is all well and good. You can get on board, and as one of the reviewers very vehemently said, he could totally understand that this is a memory molecule and that you could store information in a single molecule, but that doesn't explain how you could read this. How could the code be read? So I just want to have a couple of minutes just to highlight a few ideas of this. And this is the reimagination of the tailing code we discussed here. Apart from talking about it in terms of a binary string, folded, 4 folders, 000. So the readout is the actual proteins recruited to that site. And the hypothesis states that the number of tailing molecules in a neuron is probably quite fixed, and they're distributed across the whole of the cell in all of the synapses. So there's a finite number of each of these switches. So the relative amount of the switch in each state, in each synapse, will determine where these soluble diffusible molecules reside. So as you change the shape of this, each time you generate contractility, you change the information in that molecule, and which is read out by the proteins which are now decorated on that molecule. And something really important to point out is if you pull on a molecule where one of the domains is already unfolded, you can't re-unfold that same domain. So if you pull on that tether, then the only thing you can do is to unfold another domain. So you can have this change in these patterns depending on what state that molecule is already in at the start, if that makes sense. So in terms of cell migration, then this contractility dependent growth of adhesions is really well established. You apply contraction onto an adhesion, contractility on an adhesion, it grows, it recruits more molecules. You don't stimulate it and it atrophies and shrinks. And this would be the mechanism for how this would be working in the synapses. If you're stimulating a synapse, the adhesion would grow and the signaling molecules recruited would be different, and you'd get a stronger signaling. And you would redistribute the ligands across the whole of the synapse, which I'll show on the next slide. So what this would mean is that a stimulated adhesion would have a different pattern of folded and unfolded domains to a unstimulated. So the relative distribution of all of the signaling molecules would be different depending on the pattern and whether that synapse has been stimulated. So what I'm trying to do from here is just to highlight there's lots of things known from what I can gather. So I'm new to the neuroscience field. But there's lots of things like synaptic tagging and synaptic capture and probability of vesicular release. And all of these are known to be sophisticated and involved in these processes, but the precise mechanism is quite hard to pin down on what's going on. But one possibility, and this is quite speculative, but just to suggest the utility of this as a framework, is that the hypothesis would be that a stimulated synapse, you could have a contraction, again, sticking with this same format, these arrows, this this shows a cytoskeletal contraction. You could have a neuron where it triggers one of these, it pulls on this synapse, causing a conformational change, which causes this synapse to fire compared to this synapse where the contraction wasn't there. And that would cause a conformational change in one of these switches and change the signaling and change the release. So this could be a mechanical basis, because it's very clear from the recent papers on this that actin contractility is essential for vesicular release. It might be that it's via this change in this mechanical coding. But something which is better understood as a concept but not understood as a mechanism is that when you do stimulate a synapse, you start to get this thing called synaptic tagging, where that synapse is marked in some way in that it recruits specific molecules to that site compared to other sites. And in that scenario, you get, it could easily be explained from this. If you have a switch in that synapse, which is unfolded, you create a new binding site for a ligand, then only the stimulated synapses would have that tag, that exposed binding site. So you'd recruit more of this blue protein to that site and relative to the unstimulated. So these synapses would be talking to each other. This pool of proteins would be distributed differently across the whole neuron, depending on which synapses were most active. And another thing is inverse synaptic tagging, where you have things which seem to dampen the potentiation of the synapse. And the same thing could happen here. If they're on the unfolded state, then they could be released by stimulation and then go and sit and occupy unstimulated ones. So you can have this constant crosstalk between these different synapses to give rise to this. Okay, I'll try and wrap this up. And then building this one step further, as these switches, the history of them is locked in. Like if you've already got one pattern, then a pull on it will change the pattern again, built on that previous pattern. Then the pattern of switches in the synapse and in the neuron and in the whole circuits would be constantly updated based on what the patterns already were and what those adhesive complexes and what that synapse already looked like, which again, I think is pretty well established. But here it's a physical biochemical basis for how that might work. And yeah, I need to get a move on. So what this leads to is it leads to a model where if all of these synapses are constantly changing these patterns of switches, which coordinates their activity and the information, that if you have a circuit, this is just a textbook example of a reverberating circuit, but I've put on these MeshCODE patterns, then you could imagine a scenario where you stimulate a circuit and it causes the synapse to fire, which updates the patterns in both the pre and the post-synaptic side, which causes all of the neurons to fire. Like this is well established that this happens, but quite how and which ones fire, I would say, is less well established. But here you'd be constantly changing these patterns, and it would only be when the whole thing balances back out, all the mechanical forces sort of cancel out and become balanced again, would this stop. But once it did stop, then you would have a circuit where all of the synapses would be updated with this new information. And it might be that a future input would immediately trigger the correct output if it's learning, or it might constantly be updating this information in these circuits. So this gives a framework for you could imagine that these binary, all of this inputs into an organism are mediated through sensory, some sort of sensation, which triggers an action potential spike train, which sends that information inwards through these circuitry, through these nerves to the brain. In this model, you could begin to imagine that every single synapse and every single connection here would be constantly being updated with new information as this changed. It would change each individual synapse in a highly dynamic fashion. And then that would mean that the organism would be constantly learning and updating for the best probabilities based on the future signals. And the reason this could work is that because this is a binary code, these spike trains of a binary signal would just be updating this register. So I'm not saying I understand this, but just as a framework or as a theoretical concept of this. How long have I got left? Have I got to stop? If not, I've got about.
If this is your conclusion slide, then
it is.
We're set. Okay.
Yeah. Well, no, it's not actually. Let's skip through that. Okay, so I'll skip through all of that. I just want to wrap it up because there's a lot of possible framework theoretical conclusions from this, you could start to imagine an explanation, a mechanical explanation for many different things. And just to ground this back on our main research in the lab, is that all of this comes because we've got these molecules which have got molecular memory, and the shape of them will determine how they behave, how the cell behaves, how the tissue behaves, and here how the organism behaves. And that it can store this information persistently as a binary string. And because it's based on really well-established biophysical principles, I think that this has identified a physical location for data storage in humans and in all animals. And as I say, I think this also hints that this is a machine code. All of the cells are running the same mechanical code, which talks to the genomic code to make them all work together as a unit rather than a load of individual cells. And my argument is that this mechanical layer, and the other important thing to say here is that so far this is completely, it complements all existing frameworks for neurofunction. And I would say that the mechanical layer would integrate with the well-established chemical and ionic signaling layers to provide an underlying mechanism for information processing and storage. And I would also argue that by converting sensory and temporal inputs into a binary format, this might identify an addressable read-write memory system. So my argument, and what I think we are now starting to see, is that this is a binary code which all animals run, and that this supports the view of the mind as an information processing system or some sort of organic supercomputer. And I'd just like to finish by thanking all the people who have been involved with this, in particular Chi and Ming Chi in Singapore. This has been fantastic. All of the stuff we've done has uncovered stuff which you'd never previously have ever imagined. And it's a great ongoing collaboration with some good friends. Martin Schwartz at Yale and Nick Brown, who we've had a lot of discussions about the tailing code and how to test it. And the guys in the lab here at Kent who are helping us to test it. And thank you all for listening. And sorry I've gone over time. I had about another 10 minutes left.
That's okay. Thank you very much for your talk, Ben. That was excellent and very, I guess, thought-provoking would be a mild understatement. But I just want to open up the floor to questions. Nothing's been posted in the chat, so if anyone wants to raise a question, I think several people have to leave because of the time.
Yes.
But no, that's okay. We've got a question from Nolan. And let's start with Nolan, and then we'll go to Owen.
Hi, great talk. What happens if you just take all of the different switches and change them into one switch, so that you have the same sort of length of tailing molecule, but you don't have the information code anymore? And then you do things like put them onto different substrates. Do they still differentiate into different types of cells?
Yeah. No, that's an excellent question. It's something which we're very interested in as well. Because one of the things which we're trying to do at the minute is we can soften and harden these switches so that they go in different orders, and we can make it mimic more a soft substrate or a stiff substrate. So we're trying to see whether we could duke the mechanosensing machinery to cause it to differentiate differently as a function by changing the senses rather than the environment. So if you had them all the same, then yes, I don't know, it'd be interesting to try.
Because it'd be interesting to have, all the same of different switches, and then see whether or not you drive certain types of information systems by just controlling. It might give you a chance to understand what each of these things does.
Yes. So one of the things I took the slide out is that we can, as I say, we can soften or harden them, and we can also stop them doing their job. Like we could put a mutant in, stop it doing its role. Yeah. So it's just there as a spacer. If we put loads in, then they would all unfold individually. So you'd probably just, and this is speculation, but I'd imagine you'd end up with a dosed response. So like you'd unfold 1 and then 2 and then 3. So you'd get an accumulation. So yes, I think trying to get at this and trying to unpick this and that's a good suggestion, I would say. Yeah.
We can talk later. Brilliant.
Okay, we have a question from Owen. Owen, are you there?
Yes, yeah. I think my question was actually very related. I was just thinking that coils, so you could like favor their folding or unfolding by targeting mutagenesis to them, right, rather than relying on some external force, like to favour the switch being open or favour the switch being closed.
Yes.
And if that might be sufficient to say, like the open position favours this direction and the closed position favours that direction.
Yes, no, definitely, because most of these are the reason that they've got this, seem to have this beautiful response spin to fold and unfold so robust, is because in the core they've got, in the hydrophobic core, there's a couple of serine or threonine residues which enable it somehow, give it some sort of favourability where it can refold. If you swap those to hydrophobic, so you can really change whether they unfold or what force they unfold. And if you put serines or threonines in to other domains, you can try and make them unfold much easier. And you can even make them so that they're always unfolded. And it turns out it's very easy. It's very easy to screw this up. Like the balance of the serines inside the core is quite hard to actually do yourself. So yeah, you can make it so that you could actually fix it as a pattern of ones and O's where you know exactly what they are. Particularly if you stop it having if you uncouple it from the fourth generation machinery, you can just have it as a string of information. That's one of the things which we're trying to do as well. If that answers the question.
Yeah, thank you.
Okay, I don't see any more hands, but I just have a really